And now let's talk briefly about how scientists investigate rare hereditary disorders. And to do this, we're going to use an example of a very rare disorder called fibrodysplasia ossificans progressiva, or FOP for short. And just a bit of background on the specific disorder, FOP is a disorder in which muscle tissue and connective tissue, such as tendons and ligaments, are gradually replaced by bone, a process called ossification. This is an extremely rare disorder, being diagnosed in only about 900 people worldwide. And so how would we go about identifying the specific genetic variant that causes FOP? And just as an interesting aside, if you are in the Philly area, you can learn a lot more about this and other genetic disorders at the Mutter Museum on 22nd Street in Philadelphia. Remember that we use NGS sequencing to find rare genetic variants with large effects. A quick breakdown of this type of analysis involves sequencing FOP patient DNA using NGS sequencing. This gives us a readout of the sequence of the patient's DNA. We then take this readout and use special programs called DNA sequence aligners to compare it with the reference genome. The reference genome is the genome that was sequenced during the Human Genome Project. So this is often used for comparison when doing these types of analyses. Once the patient's DNA is aligned to the reference, we have special programs called variant callers that can quickly identify where the patient DNA differs from the reference. We can see one such difference circled in red for us. A change from a G allele to an A allele in the patient DNA. We then look closer to identify which genetic changes might be causing the specific disease. For example, for a disorder impacting the formation of bone, like FOP, we may want to start with DNA differences that occur within genes involved in bone formation and growth. When looking at genes involved in bone formation and growth, we notice a single base pair change in a gene called ACVR1. This gene functions in a process known as bone morphogenesis, or in much simpler terms, bone formation. We can see that when we align our FOP DNA to the reference, that the G allele is changed to an A allele, which causes the translation of a histidine amino acid instead of an arginine amino acid residue. This single base pair change causes the bone formation pathway to be hyperactive, leading to increased bone formation and debilitating FOP disorder.